Bacterial counting, quick, easy, and accurate. So it's time to get small. Okay, so brief overview, a little bit of introduction to what is microbial loop, why are bacteria important, little coverage of image analysis, the methods, what is macro coding, how we can actually repeat the function of counting and sizing bacteria, so results, human data versus automated data, and the time differences that we expect to get, as some conclusions, advantages versus the disadvantages of the systems and other applications that we can apply it to. So, the microbial loop. So, the microbial loop is the cyclic interaction of the microbial component of aquatic ecosystems. So, it's the trophic linkages between the bacteria, your primary producers, your phytoplankton, your ciliates, your flagellates, your microzooplankton, your mesozooplankton, and it revolves around the heterotrophic bacteria. Now, the bacteria actually recycle all the nutrients within the system. Without them, the nutrients would be lost. So all the nutrients that the phytoplankton actually produce, primary productivity, when they are fed upon, when they die, the bacteria decomposes and anabolically actually build into their systems, and the higher trophic levels actually feed off the bacteria. So the general trophic pyramid that everybody taught at school kind of looks like this. But what you don't actually realize is that everything links to the decomposers. Everything links to the bacteria, right? And the bacteria, they're found everywhere. They're found in the primary producers, they're found at the top carnivores, they're found in the middle, they're found on the sides. It's everywhere. And without them, there would be no pyramid. There would be no food webs. So traditionally, when we actually count bacteria, you either enlarge it on a photo, or you actually have to count them physically on the screen or through the microscope. Now, we've actually invented now nucleic stains that actually shine them nice and bright and blue, so you can see in the background. And with personal computers and with image analysis software that have actually come about, there's quite a lot of them out there. But even with them, a study done back in 2011 by Edward, and just recognition errors, an interlaboratory experiment, and just counting the cells that are greater than 0.7 micron, there was a 53% recognition error in just counting them. Not sizing them, just what is this a cell? And 53% from the actual microbial lab, that's quite high. So of the free softwares that are out there, the first one is called Cell Profiler. So this is found in over 3,000 published articles. It's headed by Ann Carpenter and the Whitehead Institute of USA, and it works on like pipelines. So you've got image 1, you do something to it, goes to image 2, do something to it, goes to image 3. Unfortunately, as the title says, Cell Profiler, it only gives you the profile of the cells. The next one is called Cell C Counting, written by Jariki Salumini for the Institute of Systematics Biology in Seattle. And as this says, it's Cell C Counting. Now, it can do a little bit of sizing, but the issue is with the actual calibration of the system. And I was actually looking at a lot of these journal articles. I found two that are published by the same author, 2013, 2015, and I contacted him and I go, please explain to me how you managed to calibrate the system. And his reply, and I quote, I'm not sure how well I calibrated the analysis for size. This is from peer-reviewed published work. So how we actually managed to do that, I have not a clue. The third one, which is up and coming, is from the Waholby lab, headed by Carolina, from the Uppsala University in Sweden, and they're actually doing some quite good work on algorithms, where they can segment and track analysis of actual live bacteria within aquatic ecosystems. Now, the limitations of the freeware is that it's on-point functionality. It does what it's programmed to do, that's it, it's no leeway. Other thing is, a lot of them is outdated software and hardware. There's limited or no technical support. A lot of the other times the website is closed down, the domain's not inaccessible, the author's passed along, he's moved to another university, or he's 6 feet under. So image analysis is based on binary segmentation. So binary, you take your color image and you binarize it, so two primary colors, black and white. And then you segment the object of interest from the background. So here we're actually going to segment the blue bacteria from the blue background once we binarize it. So this was originally automated back in 1981 by Cranbeck, and instead of using freeware, we're actually going to be using the commercially available software called Image Pro Plus. Now, you get all your technical support, it's really, really user-friendly, and it can be used for a vast majority of applications. And what I did, I just macro scripted some instructions. So instead of you doing this manually by yourself, it does it all together. It does it at once. So Generally, when you take pictures of bacteria, right, you're working at very small scales. So it's not always evenly flat. So if you imagine you've got a nice big hills and valleys. So when you're taking your first lot of picture, the one in the top left-hand corner is going to be in focus, but everything else is completely out of focus. You've got to continuously focus down, take a picture, focus, take a picture, focus, take a picture, fast enough that your fluorescence doesn't actually diminish. And eventually you have to put these all together to one focused image. Instead of sitting there going, file, add picture 1, file, add picture 2, file, add picture 3, I wrote this and it does it in about 20 seconds for about 300 images. It saves a hell of a lot of time. The second thing, again, instead of manually doing this by counting, file, select image, convert to grayscale, set your calibration setting. You wrote a macro and it does it all at once. So all it pretty much does, it takes your color image, converts it to gray, assigns a calibration setting, right, and then counts it. So the calibration setting we've got here is there, 77. Okay, so everything that is in red is being counted. But there's quite a lot of few cells which you can't see down there that are not being counted. This is where the user comes in. Every image for when you work with bacteria are slightly different based on your environmental parameters when you took it, how long they've been in storage. So this value does need to change slightly. So you change this image based on this histogram. So that big peak on the left, that's the actual background noise, the background data, your bit of pixels, and everything on the right is your actual physical data. Now it's the user's choice as to where you've got to align that. So here we have 67. So the more you lower that number, the more the background you're actually going to analyze. And the higher that number, the more the actual sample you're going to analyze. So if you look at these two, they're exactly the same cell, just the two different segmentation values. So if you look at number 67, you can see all the additional space that's actually around the cell that's being counted. So you're not actually although you're counting the cell, you're not very accurate in determining its biomass. And you can see at the segmentation value, you're counting over 1,844 objects. Okay, so all we do is we increase the segmentation, so we reduce the amount of background that we count, and we actually make it more accurate, which is the 87. You can see just how much more accurate you're actually estimating your biovolume. And you reduce your counts from 1,844 down to 123, and just from 20 data units. So how we actually tested the system, we got 8 volunteers, and we gave them each 60 repeated random bacterial images from all my time doing this. We trained them up into actually how to manually analyze the data. So they sat there and they manually measured the lengths and the widths of every object that they deemed as a bacterial cell. After this, they got a 2 to 3 day break. And again, from the original pool of 60, we gave them 10 images, which they recounted to test their accuracy and their reproducibility. Directly after this, we ran them through a bit of a how to work the actual macro and RPP, and they ran the original 60 again. And what we found absolutely no significant difference between the human versus automated data for numbers and biomass, which is actually what we need when we're testing bacteria. We did find a mean time reduction of total time spent analyzing of 1,136%, and a total time spent counting of 822% among all the 8 volunteers. Now, what this equates down to in real time, if you were to sit there and do nothing else, not even breathe, it's 5 hours versus 26 minutes, and a real time of 2 days versus 2 hours. Now, that's quite a bit. The surprising outcome, though, colorblindness. Okay, one of our volunteers was actually colorblind, we found out eventually, and this had a significant impact on the segmentation selection. All right, so the segmentation is you actually segment the background from the object, and he selected over 86% of his images using segmentation 47 or less, compared to all the other volunteers, and the next closest was about 63. So this had a big impact. Although his count data was accurate, his by volume estimations, because he selected so much more around the cell, was way out there, but still okay. I don't know how to really put that, yeah. Okay, so some conclusions. The disadvantages of manual analysis, it is very slow. You've got to zoom in very close and become very narrow-minded when you're counting these cells. And because of that, you actually become very strict with yourself. And you go, well, a nice blue cell when you're looking up there looks very, very different when you zoomed into 200%. You can miss cells entirely, and you're actually sitting there going, am I counting a cell? The disadvantage of the automated system, some images do require a little bit of editing. You've got a lot of detritus. It is non-specific. And you've got this do accurate count versus accurate biomass, depending on your segmentation selection. And again, are you counting individual cells? But the advantages of the automated system far outweigh that of the manual. Advantages of the manual, you know what you're physically counting, and you can judge each cell individually. But the automated system, it's fast, it's relatively easy, it's very reproducible, it counts what it tells you to count, it's non-specific, and it reduces the influence of the halo effect. Now, the halo effect is because the bacteria are shining brightly. It adds an additional angel's halo, a bit of light around the cell that doesn't actually exist. It just adds additional biovolume onto the cell. So using this automated system reduces this completely. But what does this mean for conservation? We need bacteria to recycle all our nutrients. Without them, we'd run out of nutrients eons ago, and we'd only get new nutrients when volcanoes erupted. So it would sink to the bottom of the floor and nothing would ever happen. So everything links to the bacteria. The bacteria recycle, remineralize all of our nutrients. So what we've done as humans, we've actually found a way to use this. We use them to recycle our water. So at wastewater treatment plants, we aerate the water so they grow very rapidly, and it leaves us with a bunch of sludge and cleaner water that we then clean. But at certain times, wastewater treatment plants, they get a lot of influx of water, and before they actually put that into the environment, they need to test it, either biological oxygen demand or chemical oxygen demand. Now, the BOD, or the biological oxygen demand, usually takes up to 20 days to test, and by that time, the reservoirs are full and you've got all overflowing. Now, your COD, or your chemical oxygen demand, takes a lot less time, but it requires very strong oxidizing chemicals. Now, using this counting system, you can count a sample within like an hour or two, very, very rapidly, to prevent this massive fish scales due to anoxic events caused by dumping raw sewage into river systems. So, very handy. So, other things of conservation, we actually use all these macros, which are being currently applied in our lab. We use it for mullet, which sits about there on the food scale. Sea urchins, which is a keystone species in our environments, we use them on mussels, and also can actually test biofilm growth from the bacteria. All these macros are also currently being applied for my PhD on abalone aquaculture for health and safety, as well as the Cozy Bay ecosystem, actually determining what is out there. So also, I'm also attempting to write some new macros to actually track and trace regular-tooth shark fingerprinting markings. You can also use it to assess bacterial loadings of rivers, of estuaries, of the oceans. You can also test the landfill leachate assessments. Also general water quality status, blue flag status of our beaches, as well as for microplastic counting and sizing, which you'll hear all about tomorrow. So in essence, can computers count bacteria? Well, yes, they can. They can do it simpler, they can do it better, and they can do it faster. Thank you.